Thank you, everybody, for coming to my Tech Theater talk. I'm glad that you're here. Before I even get started talking about new regulatory changes that impact your medical device and how we can assess the safety of these devices, I have to say up front, this is like a pretty large and nebulous topic. I certainly could stand here and just like drown you with information for hours and hours or days. And because it's such a big topic, we also hold 3-day seminars on this topic and covering sterilization and things like that. Our next one that we're doing is in January in Salt Lake City. There's some flyers in the back if you're like looking for a little bit broader view of what we're going to be talking about today, including sterilization, validations, and packaging, reprocessing, and things like that. Additionally, if you have any like acute questions that you want to talk about, we have a booth here. It's number 820, I think. It's on the 800 aisle. I'll be there the rest of the day and tomorrow, so we can chat about whatever you want. Okay. Cool. Okay, so let's get started. Just first, just to give just a very brief personal introduction. My name's Matt Jorgensen. I basically have been a nerd my whole life. Grew up under a rock. Didn't really get out in the sunlight very much. Stayed inside, reading books forever, making like science experiments, at home that just infuriated my parents. It's been basically my whole life. I went to school for chemistry, so I got a BS in chemistry and then a PhD in physical chemistry. Spent most of my time in academics, teaching chemistry for a while at the university level, and then we lived in Germany doing some solid state materials research before coming to Nielsen Labs. And I've been at Nielsen Labs for 3 years, heading up their extractables and leachables chemistry program there as in their consulting group. And a lot of this intersects with biocompatibility and how you need to demonstrate that your device is safe to the FDA. So it's been really great. But my one claim to fame is that I helped to invent a flavor of Mountain Dew. This is Mountain Dew Whiteout that you see there. It's a smooth, citrus, refreshing Mountain Dew flavor that I think is still on the market now, 10 years later. Because I helped to create this flavor, they put me in a Mountain Dew commercial, and for that reason, people call me Dr. Dew, and you all are also welcome to call me Dr. Dew if you would like. And this is really my one true claim to fame. But what do we? So regulations, changes, and how that affects your medical devices. So there's been a lot of changes on this regulatory landscape recently that are kind of dramatic, right? So, for example, the new ISO 10993-1 just came out in August. We've been awaiting this new draft for a long time. The previous draft has been out for decades. We knew that some of these changes were coming now. These are really official, and here I'm going to talk about that in detail. There's the new ISO 18562 document that came out March of last year. This affects all gas path devices and how you need to assess the safety of those. The new MDRs have been out and ratified. That the next major deadline is May 2020, so this date is rapidly approaching. If you're not thinking about becoming compliant with the MDRs and you have medical devices on the market or for market in Europe, then this is something that you need to be thinking about right now. And then, of course, the 10993-18. This is the ISO 10993 document that focuses on how you should conduct chemistry testing. This has been in draft and redraft, and we think that we're finally very close to getting this finalized. So that's coming. So I'm going to spend most of my time talking about the new ISO 10993-1, and because I'm a chemist, I will talk a little bit about chemistry, and because this is like the most major change. So many of you should have seen this document. This is the FDA guidance on the application of 10993-1. And this was issued in June 2016. And to kind of like put things into perspective about how fast things are changing, This document replaced the old G95-1, which stood for decades. It was the only document that we had from the FDA on biocompatibility. So in 2016, they released a new one where they list out all of these biological risks that they want to be addressed, and there's X's and O's, giving you guidance on whether or not you should do that. Now we have the 1099-3-1, the new one, which more closely mimics or mirrors what we saw in the new FDA guidance document. And let's just sit and kind of like digest this picture a little bit. So this table that you see there is table A1 from the annex of 1099-3-1. And if you looked at the previous version was a little bit more like this one, where you had X's. And the title of the table was biological tests to be considered or to be done. And we got rid of the X's completely and replaced those with E's. Now, this might seem like a minor change, just like, changing like the letter used from an X to an E, but it's really a little bit more significant than that, because it officially shows this paradigm shift where we're moving away from Xing out or checkboxing a set of tests that need to be completed to an evaluation method where we say, okay, rather than do these tests, you need to consider and evaluate all of those risks. And so now we're in a place, and we were doing this over the last couple of years leading up to the release of this document. What this really means is that for a certain type of device, you can pick any of those categories that you want. Just because there's an E in those columns, it doesn't mean that you need to test that. It just means that you need to address it or evaluate it. And we found that there's lots of situations where we can address or evaluate a biological risk with no testing, maybe by leveraging the clinical history, the use of the device and the materials, or other routes to address it. So these things just need to be evaluated, not necessarily tested. And if you're going to be evaluating your device, you kind of need to understand what's in it, the materials and the chemistry and how it interacts with the body and everything like that. So it's really more of a comprehensive, risk-based approach to evaluating biocompatibility that leverages your understanding of the device and its materials. So I'll talk a little bit more about how that works, but this is a pretty significant change that we have there in the pipeline. So all of this is coming together as an encompassing strategy where it's all about risk. We're talking about using a risk-based approach. This language about risk is throughout the FDA's document and the new 1099-3-1. You can see in the FDA's document, they spell it out explicitly that as you evaluate biocompatibility, you should be thinking about the material components, manufacturing processes, previous clinical use, and you evaluate potential risks from a biocompatibility perspective, and then you evaluate those either using testing or other evaluations that appropriately identify the risk. And the feeling that we've had from our European regulators especially, and now it's moving more and more into the FDA, that they really expect you to evaluate as a first option as opposed to doing any animal tests. So if you are going to do an animal test, you should be able to justify why you had to do the animal test rather than just defaulting and moving to that in the first place. So this is really kind of a different direction there. So the way that we take care of this at Nelson Labs, and this should be kind of more of a general approach, if you're going to be evaluating the biocompatibility of your device, it really follows a natural 3 key steps. And these are outlined in the FDA's guidance document as well. So first, you should start with a biological evaluation plan. And this seems to me to be kind of obvious, but you're so anxious to get started with testing and stuff. Sometimes people stop and they kind of forget to make a clear and concise plan about what are they trying to accomplish. And in this biological evaluation plan, there should be a written document that's auditable that you could give the FDA, where you describe the device, what the device is made out of, the materials of construction, all of the processing, the biological risks which need to be evaluated, and then what you're going to do to evaluate them. What I would highly recommend in this regulatory environment that we are now that's changing very rapidly is that if you're going to start down this path where you need to evaluate the biocompatibility of a device that you want to get to market, you really, really have to make one of these plans. And I would strongly recommend that you show this plan to the FDA or whatever regulatory body that you're submitting to, so that they can give their nod of approval on that before you start down this very long and expensive path of evaluating the biocompatibility of your device. And within this plan, if you feel like using a strategy that's maybe a little less traditional, if you're going to evaluate a bunch of endpoints with just written assessment alone, you can say that you're going to do that in the plan, and the FDA will let you know, okay, yes, we'll accept a written assessment, or no, we'd really prefer if you did chemistry or some other tests. So that's really the first step, is making that plan. Once you have the plan, then you go into the testing and evaluation or assessment phase. And then finally at the end is this sort of capstone report that summarizes the results of all the testing that you did. So in this biological evaluation plan, you should go through a process where you identify the risks associated with the device and try to determine if you need to do testing. And this flow chart is coming from the new 10993-1. And here you can see there's all of these checks or questions that you can ask yourself to make sure that you really need to do animal testing. So the first one here is the material the same as one that's commercially available? And the next question is, does the device have the same chemical composition as something on the market? And so on and so forth. And if you're following this path and you can answer yes to all of these questions, then you don't need to do so much testing, right? So that's exactly what we do. Material characterization is a big part of this biological evaluation plan, and if it's not in that plan, it should be documented and included and auditable. So according to 10993-1, the selection of the materials used in the device first need to be selected based on their fitness for purpose and regards the chemical, toxicological, physical, and so on properties of the material. So you need to demonstrate up front that you've selected your materials thoughtfully, not just from a functionality perspective, but from a toxicological perspective down the road. And if you are very careful and have good information on the front end here about all of your materials, it can really save you a lot of time and money down the road when it comes to testing. So that's what goes into the biological evaluation plan. If you prepare one of these, or if we help you prepare one of these, which we're happy to do, we go through the material characterization, we talk about the device and how it's used, its intended population, all of that. We explain testing families, like family groupings, if you're going to use some sort of representative sample or coupon to represent a range of materials or configurations. And then we outline the actual tests that are recommended or that will be executed. So I thought we could just maybe go through the thinking on one of these just as an example. So consider if you have something like a dental aligner. So like one of these trays that adjusts the position of your teeth in your mouth. So traditionally, like say 3 or 4 years ago and before, this is a type of device that might not have any biocompatibility testing at all. They just didn't really feel like it was necessary because it's in the mouth. But now we're testing these all the time, so I see quite a few aligners across my desk. So the type of device that this would be, even though you take it out every night, the cumulative exposure is definitely more than 30 days. So we say this is permanent, and it's in the mouth. So we would call this permanently contacting with mucosal membranes, and you can see all of the endpoints that you should address are labeled with ease there. And I'll just point out right away that even though we spent years and years working on the new 10993-1 in the finalized printed version, they did miss the heading of one of the columns there. The one that's blank should be sensitization, so that still is a required test. It's just not labeled there on the document. And one of the things, so those are the endpoints that have to be evaluated. And if we were to start to make a plan like this, and if you were just like call me up on the phone and be like, okay, well, what should we do, Matt? And I always say, well, you have options, right? There really is no one way only that you can do this. You have multiple paths to regulatory success here, right? And for any of those endpoints that need to be evaluated, you certainly you could try to do no testing where you defend the safety of the device just by some written document, or you can do the traditional test, though if you could be challenged on that later, right? If you could have done something easier to spare animal life, or you can do chemistry testing followed by some sort of written evaluation. And so what we would do is we would go down this list and we'd say, okay, cytotoxicity testing, this is really inexpensive, it's pretty fast, it doesn't use animals, and the FDA does like to see it, so we'll basically always recommend the traditional test for cytotoxicity. For sensitization and irritation, we either always do the traditional test or just a written evaluation. And the written evaluation we found works really well if you have a very common, well-characterized material. So you can think of something like a well-known titanium alloy or a stainless steel alloy. We use this strategy a lot. So if you have something that's like a 316 stainless steel, this has been implanted in people for decades and decades. It's been done thousands and thousands of times. It's also been tested thousands of times, and it's we know that 316 stainless steel is safe. That's not a question. The only risks associated with a really common material like that is if there's any residuals on the device due to just the manufacturing environment. So if you have a very common material and you can prove somehow that the device is clean or free from residual manufacturing materials, then you can often avoid these tests straight away. For something like a dental aligner, a lot of these are like 3D printed. The materials are pretty novel. We usually don't know enough about the materials or have enough experience to avoid these tests that way. So we would recommend sensitization and irritation, the traditional testing there. Then for the remaining systemic toxicities, really any of these could work. If you have a very common material, it's the same argument as before. You can do the traditional testing, though I can say that acute systemic's not that bad, but subacute and subchronic testing together with chronic tox is a pretty expensive, long endeavor. You're looking at at least $100,000 in a few months. So we try to avoid that if we can. So what we would recommend by default is chemistry testing, followed by some sort of toxicological risk assessment in this case. And then here, for something like a dental aligner, I'm sure that we would recommend just having a written evaluation based off of the material and how it's used. Implantation testing really evaluates the local reaction of tissue with the device material. Since this is mainly touching the teeth, it's not in permanent contact with tissue all the time. I think that we could explain why implantation isn't a real risk. And then in something like a BEP, we would make a table like this. We'd say, okay, FDA or whoever we're submitting to, here's the endpoints that need to be evaluated per 10993. Here's what we're going to do. We'd submit a Q-sub or a pre-sub with the FDA. We'd get them on the phone and we'd say, okay, well, what do you think of our plan? They'd say, well, yeah, it's good, but we want some other crazy thing. Or they'd say, yeah, it's good and you're fine. And then you go forward, you have a record of that, you do the testing, and you're set. Yeah? Makes sense, right? I think it's great. So this middle one there, the chemistry testing and assessment. So, of course, I'm a chemist, so this is where I get like really, really excited, and everybody else is kind of like falling asleep or whatever. But this chemistry testing is something that we've been using to support biocompatibility now for a few years. Even though we've been using it and it's been really, really well accepted, I would say it's also one of these areas that's nebulous in the sense that we see changing requirements from the FDA all the time. The feedback that I get from the FDA today is different than what it was 3 months ago and 6 months ago and a year ago, and we're constantly trying to adapt to meet what the FDA wants. And I think that what's going on there is that the FDA is understanding the value of chemistry for evaluating the safety of medical devices, and they understand also that the details of how you conduct chemistry testing really, really matters in terms of quality of the result. So they've hired a bunch of chemists, on, and they're really scrutinizing the way this is done. So, I'm just going to talk a little bit, not too much about chemistry, but a little bit about chemistry just to get everybody excited, and then, try to reinforce that the details of how you do this study really do matter, and that you could be scrutinized by the FDA for how you do it. And so the type of chemistry that we do to support biocompatibility of a medical device as a substitute for subacute, subchronic, genotox, and those other systemic endpoints is ENL. We call it ENL. And before I get in trouble with my group, they always say, well, you need to define what ENL is. So when I say ENL, I mean extractables and leachables chemistry testing. This kind of has a special meaning within biocompatibility. These are terms that we're borrowing from the pharma world. Extractables are chemicals that can come off of a device or leave device materials when exaggerated, aggressive conditions are used, things that we know are going to be worst case compared to clinical use. And then leachables are what we expect during actual clinical use in contact with the body. So extractables, we use something like hexane, which is a pretty aggressive solvent at 50 degrees C to elevated temperature and for 3 days. And whatever comes out under those conditions, we'll say, okay, well, this is the amount that could come off every day for the rest of the patient's life. So it's a really exaggerated scenario. If we went to leachables, we do something like saline for 37 degrees C and 24 hours. So that's really the key difference. And I think the reasons why you should consider using chemistry testing are pretty clear as an alternative to those biocompatibility tests. You save a lot of time and money, right up front because it's a lot less expensive than running those bigger tests. You get really detailed results. So if you do a subacute or subchronic animal test and you fail, then all you know is that you failed and that you're in trouble, right? But if you do the chemistry testing and there's a problem, then you know what that problem is. It's localized to a certain chemical that you can then trace back and try to understand where it came from. It gives you a roadmap to solving the problem. And then, of course, the FDA is just really strongly recommending chemistry testing in cases where devices are in permanent or prolonged contact with the body. So it might be that even if you do all the animal testing and pass all the animal testing, the FDA still could say, well, wait a second, we still want to see chemistry testing. So we've seen this a few times as well. So the reason why we do chemistry testing, the ENL for biocompatibility, I think it's best summarized by kind of like an embarrassing, personal story. So, in addition to loving Mountain Dew really, really a lot, I also really, really love pizza. And, I love pizza so much. I had to plan this special trip to Naples in Italy so I could have pizza where it was really invented, right? And we sampled all these famous pizzerias like the Pizzeria Sorbillo and so on. And, I spent all my money on pizza and, travel. I didn't really save very much for the hotel. So I stayed in a really, really crappy hotel. And it was kind of crappy to the level that it was pretty dirty also. But on the last day, they finally cleaned the bathtub. And I was like, oh, so what a relief. So I took a nice hot bath. And then when I woke up in the morning, I had this sweltering, welting, rash all over my body. It was absolutely miserable. The lady at the pharmacia thought it was hilarious. My wife was laughing her butt off, but I was just absolutely miserable. And so this makes me think about ENL chemistry testing, because if you think about the bathtub as a device, and then this cleaning procedure is sort of like the manufacturing process, then if we want to understand, does this bathtub pose a risk of adverse effects to humans, we could do what I did, we could test it out on a living thing and see if it makes it miserable, or what you could do is you could make the bathtub and clean it according to the instructions for use on the bottle, and then fill it up with hot water as it would be used, and then just take a sample of that water and test it using a variety of analytical chemistry means. And if you did that, you get a list of results. You'd probably find lead in there from old Italian pipes and all this other stuff. And then you find some organic compound that's a really strong known toxin or an irritant. You'd be like, oh, wait a second. this is not okay for somebody to be covering their entire body with in a bathtub. Where did this come from? And you can cross-check that with a list of everything that contacts the tubs and be like, oh, well, it's the cleaner, right? And, probably a good fix for that would be to, add to the manufacturing process an extra rinse cycle to remove residual cleaner, or, add it to the instructions for use, like don't use this bathtub without rinsing it out first, or whatever. But there would be a way to address it like that. So that's really the whole goal with this E&L chemistry testing, right? We want to find potentially concerning or toxic chemicals, identify them, and so that we can prevent, adverse reactions from happening. Now, when I talk about, why it's important to, which strategy that you use for your chemistry testing, this really comes into focus if you start to think about the scope of the problem here. And really what a toxicologist or the FDA would want to feel really safe and comfortable about a device, or also what you would want yourself if a device is being used on you. So a good example for this is like this. So this is a real medical device. This is a 3D printed Spock hand. Its intended use is to be implanted into the body next to the heart, so you can keep the memory of Leonard Nimoy with you always, right? Now, if you were the Star Trek fan that was receiving this medical device as an implant, and you start to ask yourself, okay, how rigorously or how carefully would I want this thing to be tested, you'd be like, okay, if Dr. Jorgensen said, well, we're going to screen this device for 4 potentially toxic metals, and, we'll throw in pesticides for free, you'd be like, okay, that will make me feel a little bit better. But, not only do I not want there to be potentially toxic metals or pesticides, but I also want to make sure that there's nothing that you don't expect is in there. So basically, the scope of the problem is we need to look for everything and anything, the things that we suspect might be a problem, and be able to catch things that we don't expect, because we know all sorts of crazy things happen in manufacturing environments, additives 1 or 2 levels upstream in material supply chains. We have no idea what actually could be in there. So the testing strategy has to be one that is screening, so that it can look for anything and everything that comes off of a medical device. So you need to cover all of these categories of chemicals and compounds that could be found, and that's exactly what we do. We have different analytical methods to look for all of those. Okay, another essential part of this whole program is that if you're going to extract a medical device in something like isopropanol or hexane or water, and then you want to understand what's in that water, there's only certain analytical techniques that are up to the job there. And if it's going to work and make this identification, one of the critical steps is that all of those compounds in the mix have to be separated from each other. So if you're using a method, say like non-volatile residuals with FTIR, it's hopeless, right? You can't identify all the different components if you don't separate them from each other. And to do that, you really need chromatography. So chromatography is an analytical chemistry technique that's used to separate different chemicals from each other. And I show some pictures here. This is like the old school way of doing chromatography, and anybody that goes through like a chemistry program has to do this, and they always say, well, back in the 60s, this is how we did chromatography, and you separate these things out on a paper, then you cut out the spot from the paper, so we could cut out just like the green part there, and then you would analyze that using a variety of methods. Of course, we have better techniques now. We have gas chromatography and liquid chromatography that are used to make this separation for us. But I'm just telling you this because if you decide to use chemistry to support the safety of your device, and you're like ordering testing, if there isn't chromatography that's part of like the quote or the mix or the plan, then it's like it has to be like a full stop, right? You're like, okay, wait a second, you're jangling my chain here or something, So it has to include chromatography. That's the separation step. It's absolutely required. There's a lot of details that go into how chromatography is done. It's a whole art in addition to a science. So it's good to find a lab that has a lot of experience doing this. So when we talk about the whole scope of everything that could come off of a device, right now, we generally think of this into like three general groups. On the organic side, we think about very light, volatile chemicals. These are, volatile organic compounds. That's one category. And what's great for that is something like headspace, gas chromatography. Then we talk about like medium-sized molecules that don't vaporize so easily but still can evaporate. Those are semi-volatile compounds. We have a GC method for that. And then for really big, heavy molecules where if you tried to evaporate them into a gas, they would just burn or break down, then we use liquid chromatography for that, so we don't need to evaporate them at all. So there's, generally should be two gas chromatography methods and a liquid chromatography method. For the metals or anything inorganic, if it has a metal in it, then we generally use like an ICP method, either an ICP with mass spectroscopy or ICP OES, optical emission spectroscopy. And if you can identify which metals are coming from your device, you can usually track down these inorganic parts. So the gas chromatography and the liquid chromatography separates. On the organic side, the identification step is also very important, and we could talk for a really long time about the art of mass spectroscopy. Once these compounds are separated, they have to be identified. We use mass spectroscopy to do that. There's lots of different flavors of mass spectroscopy. If you want to pursue this, or if you're ordering this, it's good to ask a lot of questions and also talk to a toxicologist and see which degree of certainty you need things to be identified and things like that. But this is like the essence of extractables and leachables chemistry testing for biocompatibility. And if you want to talk more about chemistry, I will be waiting very, very anxiously at the booth. If just anybody, just please talk to me, Okay, so I'm going to move on from this. I want to talk also a little bit about 18562. So if you have a gas path device, ISO 18562, you should have downloaded this, printed it off, and now read the four sections at least a couple of times. So this is really important if you have a gas path contact device. This is recognized by the FDA. If you have this type of device, it's required. And to me, so we were doing biocomp before 18562 came out, and we would have gas path devices, and we would go back and forth with the FDA, and we would do the testing, but the testing in 10993 for gas path devices really doesn't make a whole lot of sense. If you think about doing something like a cytotoxicity test, where you take the device and you expose it to cell culture fluid, and then you put that fluid on cells and see if it kills cells, that's great for something like a catheter. But if your device is like a machine with lots of complicated moving parts and lubricants and stuff that never touches the patient, and if there's highly reactive active compounds in there that are great for a gas to pass through but explode in contact with water, then it's kind of a problem, right? And so we were always struggling with this. So this, I think, is like a gift from heaven, especially if you have gas path devices, and it's really common sense. So if you ask yourself, well, okay, what's the risk associated with a gas path device? It's basically just what can contact you through the gas. And so that's what we test. So we test for volatile organics in the gas from a device or passing through the device, and we also look for particulates. And those are the two main tests for a gas path device. And if you have something like an oxygen concentrator or an oxygen tank or whatever, where the only contact through the patient is through the gas, then these are really the only two tests that you need to do. Volatile organics in the gas and particulates. The whole situation becomes a lot more simple and a lot more straightforward to address the risk. So the way that we do this, just briefly, to measure the amount of volatiles in the gas, what we do is we either direct air through. If we're talking about something like an oxygen tube, we would direct clean, dry air through the tube, and then we'd sample the output using a vacuum canister. So you run air through it, you suck the air into a canister, and then this canister is taken to a GCMS and expelled, and they analyze what's in there. For the particulates, you can do either by weight using filters, using a couple different sizes, or you can do line scattering methods. So it's actually pretty straightforward to execute. Okay, So after you do all of this chemistry testing, either on the gas or on the liquids, then you're left with this chemistry report, however that may look. And this is the report there on the left is kind of like the stopping point for a chemistry test. And now you need to kind of understand what the next step is with that. So if you're a company that has a toxicologist on staff, they might be able to take a look at that information and be like, oh, this okay plasticizer, yeah, we're not worried about that, but whoa, that bad plasticizer is concerning. Then that type of assessment needs to be made. If you just give the FDA or TÜV or DEKRA these chemistry results, they'll just laugh at you and like throw it back. They say, okay, well, you have to tell us what this means, right? Or somebody that's qualified needs to tell us what this means. And that's where a toxicological risk assessment comes in. So if you're using chemistry to address biocompatibility, you have to remember it's like a two-phase process. One is like this art and beautiful science of chemistry, and then after that is somebody qualified needs to interpret the results and make a claim about safety. So the toxicologist will say, okay, we've reviewed all of these compounds, and based off of my expertise, the risk of chronic toxicity, subacute toxicity, and so on is low, and it's actually that part that the FDA really cares about. Okay, so two guys walk into a bar. One guy orders an H2O. The second guy says, I'll have an H2O too. Of course. Okay. I love that joke. Okay, I'll just finish with a quick example. I think we just have a couple of minutes left. It's really kind of like highlights, I guess, the benefit of this new strategy, this, why we're using all E's and why we're using chemistry. And it's one that, I guess it was recent. Now it's been about a year or so. So we had a sponsor that we dealt with that had an enteral feeding tube. So this is intended to deliver nutrients to an underweight neonate, baby. And they had gone through the whole process of doing the biocompatibility test per 10993-1. They just straight, check box the test and did all the tests and passed. So they submitted this whole docket to the FDA, and the FDA said, okay, great, this is nice, and we understand that biocompatibility tests, including the ones that you conducted, are on adult animals, not underweight developing animals, and we don't really find animal tests to be adequate in this case, right? So they kicked it back, came to us, and the FDA recommended chemistry and a toxicological risk assessment. So that was kind of the setup here. So really the situation was a little bit complicated because it was like a kit. There was multiple tube materials and the delivery system with the syringe and so on. So we had this whole testing strategy. We did this chemistry testing, both extractables and then the leachables. And the end result was that we found 27 compounds and metals in total across all these different parts. In 12 cases, those compounds didn't need any evaluation at all because the concentrations were so low we could throw them out right away. They were below the threshold of toxicological concern. And then 15 compounds needed a more detailed assessment by our toxicologist. And one of those compounds of the 15 was one that was legitimately dangerous to underweight neonatal babies. So in this case, even though the FDA does get a little bit of a hard time, they are pretty smart. They do the right thing a lot of the time, and they were right in this case. These animal tests weren't adequate to protect a very low weight vulnerable population, and chemistry was the right thing to do. And had this gone to market, there's a good chance that it could have hurt underweight babies. So the path forward there was, since we had identified what this compound was and we had broken up the parts of this kit into different pieces, we knew that it came from the black rubber syringe gasket in the syringe to deliver the food. So the manufacturer was able to go back to the maker of the black rubber syringe gasket, and the manufacturer was like, oh yes, of course, we know this compound, it's a vulcanization agent, we use it to make the rubber more stiff, and yeah, we have alternatives for that, we can reduce the amount. The manufacturer of the gasket made that change. We did a nice follow-up test just to confirm that everything was in fact low and where it should be, and the FDA accepted those results, and the device is now on the market. So it's a great success in the fact that we protected a vulnerable population, and we're able to still fix the problem and get the device to market. Okay. Final slide. I promise we can't really talk about regulation changes and things like that without first saying something about the MDRs and how this also ties back to what I've been saying about 10993-1 and chemistry testing and all of that. So the MDRs are here. The first major deadline is coming. It's in May of 2020. The MDRs are pretty significant. If you have a device that's going to be marketed in Europe, even if the device has been on the market for a long time, you still need to be able to prove that the device is biocompatible. And just it being on the market for a long time won't be sufficient to address this. So it's explicit. There's no grandfathering precisions at all. Everybody has to reprove it. There's an expanded definition of what is considered a medical device, and it's explicitly required that a qualified person carries out this evaluation and assessment. So this is one of those things where if you have devices on the market in Europe and you don't have biocomp data per 10993-1, then we all should kind of like look within ourselves and within our files and see, okay, do we have biocomp data per 10993-1? If the answer is no, then we should start testing right away or figuring out how to prepare to meet this requirement as it comes up. So the MDRs are coming. If you need any help assessing or evaluating the impact of that, of course, we're here and able to help. With that, I'll just close here, and if there's any questions, I'm happy to answer them here, or we can talk one-on-one today or tomorrow here at the show. Are there any questions? Yes. Yeah, so the question was, does the new ISO standard 18562 recognize the value of filtration of the gas? Yeah, and the short answer is, yes. We love that those filters are there, but even though that there's a filter there, there's still kind of a question if there's any dust on the filter or just past the filter and things like that. So you still have to test, though it's good that those filters are there for sure. And filters are like one of these very high surface area things that people wonder about VOCs from as well. So, yeah, it's a good question. Any other thoughts? No? Okay, good. Oh, yes. Yeah. So the comment was, she's heard in the past from other webinars and things like that chemistry and evaluations were becoming more and more important. And do I have any examples where somebody has done just a straight paper evaluation, no testing, and been successful? And so the short answer is yes. This comes across my desk maybe once a month, maybe once every 2 months, where we do a project where there's no testing. No new testing at all, right? And in these cases, it's a material that's very well characterized, a metal material, and they have data like the previous cleaning validation data that demonstrates that residuals aren't an issue. So if you're in that category, let's say a nitinol stent and a well-documented cleaning validation, then that's enough. Or it has been enough in the past, right? So we've done that. Or the example I gave before, specifically at like a titanium knee and a good cleaning validation, then we've been good. So it is possible with only written evaluation. Okay, let me give you just another example. Or if it's a device that's a small change from a previous device, and the magnitude of this change is really well understood. So you change a color of a non-patient contacting part. I mean, these ones happen more regularly and are pretty easy. Yeah. Okay, great. Well, thank you for coming, everyone. I love chatting about this stuff, so come and visit with me anytime.